It's wonderful to see so many familiar faces in this room, and a lot of faces that I have yet to meet, but hopefully we'll get a chance to talk after the lecture. My name is Diana Robbins, and I'm the interim director of the A.J. Drexel Autism Institute here at Drexel University. And it's my great pleasure for the first time to be introducing our annual Autism Public Health Lecture. So as you can see that this is our 7th year, and many of you know that our founding director departed to become the Dean of the College of Health and Human Development at Penn State. And I feel like I'm doing an okay job filling his shoes so far since January, but my feet are a lot smaller, and some of you may realize that there's about 10 inches less of me than you've seen in previous years. But I'm gonna do my best to tell you about some of the exciting things that we're doing here at the Autism Institute. We were the very first autism research center to be created with the mission of public health science. And it's something that I'm incredibly proud of, and many of our team are here today. And one of the things that I was just saying this morning at breakfast is I am incredibly proud of how passionate every single member of our team is about the work that we do. And by being a public health research institute, what we really want to do is bring science to the real world today, not 10 years down the line or 20 steps removed from some of the work that we do. And so our institute is formed around 3 research programs. The Modifiable Risk Factors program, which you'll hear more about today because we rotate, and today's lecture really fits in that domain, focuses on pre and perinatal factors that might bring both risk or protective factors to the development of autism in a child. The program that I lead is called Early Detection and Intervention, and we think of this as more of a secondary prevention, whereas modifiable risk factors, if we can identify things to change in our environment, it might actually change the development of autism. In early detection and intervention, what we think about is a brain is already developing on an autism trajectory, but if we identify it and treat it very early, we can alter that trajectory and improve outcomes. And the Life Course Outcomes program is our 3rd research program, led by Dr. Paul Shattuck. And we think about tertiary prevention here. So a person has already manifested autism spectrum disorder, but what can we do to improve quality of life for the individual and their family? And some of their work specifically focuses on a very challenging time, which is the transition out of the public school period into adulthood. And so our 3 research program areas are hard at work every day, but we couldn't possibly do this work without the amazing team that we have in our institute. And so I want to just acknowledge a few people who have been particularly helpful in supporting our research programs, but also the public health lecture today. So first off, I want to thank the Outreach Corps. So Dr. Jennifer Plum, where is she? Wave. Oh, she's in the way back by our Eagles table. She leads the Outreach Corps, and they do an amazing job putting on events like today's lecture, which brings together family members, community partners, scientists. I'm probably missing 10 other types of folks who are here today, but our Outreach Corps is there to help people learn about what we do and connect us in ways that can promote the science that we do here at the Institute. And I know her team has worked incredibly hard for today. So Bushra, where are you? Give a wave. Bushra's over there. Meredith in the back. And Jamie, who I think has worked tirelessly to make everything run smoothly, and she is now doing her civic duty and serving in a jury for the next 2 weeks. So all her hard work, and she didn't even get to be here today to have a bagel and see everybody paying attention to the amazing talk we're about to see. I also want to acknowledge our clinical core, Dr. Jim Connell, who's off to the side there, and his team. They do an incredible job not only supporting some of the research that these 3 programs are engaged in, but also engaging with many community partners to bring our autism services to agencies that provide diagnosis, agencies that want to create positive work experiences for individuals with autism, and many others. We also have our admin core, and Mitch is somewhere up in the back. Mitch also helps organize and keep everything running smoothly. And I also want to acknowledge our Policy and Analytics Center, Dr. Lindsay Shea, give a wave, who is one of the things that I think is really unique, helps connect our science to actual practice by communicating with lawmakers and policymakers. And so the team that we have here, I think, is really unique, and I just want to give a quick thank you to everybody who contributes to it. So today is our 7th annual public health lecture, and I just wanted to show you some of the wide range of speakers we've had here. As I mentioned, we rotate through our different research programs. So last year was Dr. Obin Stamer, who talked to us about implementation science and early intervention. And you can keep looking back through all the years. The very first one actually was Dr. Paul Shattuck, which I guess was part of what drew him here to Drexel, because he runs the Life Course Outcomes program. So without a whole lot more, I wanted to just say that in just a moment, I actually have one more slide before we switch over to Dr. Krone. I wanted to take just a minute to talk about the Eagles Autism Challenge. I think many people in the room have already heard about it, and we actually have some partners here from the Eagles Foundation, which is really exciting. But last year was the inaugural Eagles Autism Challenge, and they worked tirelessly and raised over $2.5 million. And 3 of our junior faculty were beneficiaries of research grants from that first challenge, and they'll be getting started next week. April 1st is their start date, so that's really exciting. And I'm sure in a year or two you'll hear about some of the work that they're doing with these funds. But I wanted to, if I can do this, I wanted to show you just a really short video. How do I make this sound? Come on.
Drexel is back and participating in the 2nd annual Eagles Autism Challenge on May 18th at Lincoln Financial Field. I can't wait for you to ride, run, or walk with the Eagles team. Together, we are changing awareness into action and raising funds to advance cutting-edge autism research in the Philadelphia area. Reach out to your team captain to get started, or visit eaglesautismchallenge.org. Thank you for your partnership, and I look forward to seeing you at the finish line in May.
So for anybody who went to the challenge last year, I have put in a request for better weather. It was a little cold and rainy. It was still really, really fun, but it was a little cold and rainy. So I think it's going to be perfect weather this year. And it's a really fun time. And just from personal experience, we walked alongside like actual football players who took their Saturday to come and hang out with families and scientists, and they were totally personable, real people. I walked with the guy with the big beard, Jason Kelsey, who became super famous from the parade, and he was just a really great guy to chat with. And I think it's a great opportunity, and I hope that everybody here will think about either joining our Autism Institute team or the Drexel team, or one of your own teams that you can affiliate with Drexel to show your connection to all of us. So without further ado, Dr. Kristen Lyle is going to introduce our speaker this morning. Thank you.
Good morning. I'm really honored to have the opportunity to introduce our speaker, Dr. Elisa Cronin, today. She's truly a leader in the field of autism epidemiology. Dr. Cronin is a senior research scientist at the Division of Research of Kaiser Permanente, Northern California, where she also is director of the Autism Research Program there. She received her PhD from UC Berkeley, as well actually as her MPH and undergraduate degrees, so she's a longtime resident of beautiful Berkeley, California. Her work focuses on the epidemiology of autism and neurodevelopmental disorders, with particular attention to perinatal risk factors and immune risk factors. And she has found ways to make use of the unique resources available in California to conduct really novel studies, a number of which I'm sure we'll hear more about today, including the Early Markers of Autism study, the Autism Family Biobank, and the Impact study. She is also a key investigator on a large number of other highly impactful studies, including the National Collaborative ECHO study, which is investigating environmental influences on child health outcomes, the multi-site large population-based SEED case control study, and the early study in which she led one of the sites there, among other studies. She has served on a number of scientific advisory boards for autism, including currently the Wendy Clagg Center for Autism. She has successfully led and received NIH grant funding for numerous projects that are really collaborative and innovative in their nature. She has about 12 pages worth of scientific abstracts and presentations and over 150 publications, a number of which have been the first paper addressing a given topic and really stimulated research in new areas. And also many of which are first and senior author, which speaks not only to her productivity, but also to her strength as a mentor. Beyond this very impressive CV and what I would call rock star status in the field of autism epidemiology, she is also just someone who is really wonderful to work with. Her mix of productivity, professionalism, and strength and mentorship is, I think, really rare to find. And I personally started following Lisa's work with a landmark paper that she led sometime around 2000 that was addressing factors influencing the increase in autism prevalence in California. And after that, I don't want to say that I stalked her by email, but I just might have sent her very frequent emails asking if she had any job opportunities. And while that didn't quite pan out for me at the time, I can say that I've been lucky enough to experience her mentorship firsthand through collaboration on a number of projects. And she is someone who continues to inspire me, and I'm sure will inspire all of us with her talk today. Dr. Krone's work and the innovative studies she has led have truly advanced our understanding of perinatal risk factors, as well as mechanisms involved in the development of autism. And with that, I'm very pleased to introduce our speaker, Dr. Elisa Cronin.
Wow. Thank you so much, Kristen, for that lovely introduction. Let's see if I can put my water down somewhere. Shall we go? Okay. Do you want to? Is that it?
There you go. There's an advancer right here.
Okay.
If you'd like to use this as wireless.
Oops. There we go. Okay.
And this is wireless advancer forward and back.
Okay, great. Well, okay, so thank you so much for that wonderful introduction and inviting me to speak today at this lovely event. I have seen people way at the back of the room. There's lots of room up front if you want to come a little closer. It's just really a pleasure to be here and to get a chance to talk about some of my work that I've done over many years. And truly, this work could not be possible without collaborating with many, many individuals, Kristen Lyle being one of them. Kristen and I met, as she said, early on in her career, and I feel very fortunate to have collaborated with her as well as Brian Lee and several other people who are in the Philadelphia area on a body of work. So, and what my goal today is to talk about epidemiology and what epidemiologists do and sort of tell a story about my work through the lens of looking at immune function in autism and where that's led me, where we've come from, the studies that we've done, and some of the studies that we're currently embarking on based on all the research that led up to this point. So in overview, I'm going to talk a little bit about what is epidemiology. I'll talk a little bit more about what we know about the epidemiology of autism, and then really focus on, as I said, immune function in autism, and describe several different studies that I've been involved with, as well as other people have published in the field, and then talk about what we're doing currently. So what is an epidemiologist, anyway? So here I've put a picture of a bug. I get commonly, when I say, oh, I'm an epidemiologist, actually I don't say that anymore because people get so confused. I say I'm in public health, I do public health research, but you know, when I'm feeling brave, I say I'm an epidemiologist, and half the time when I'm talking to people in the community, you're like, oh right, you know, you study bugs. It's like, well, not quite, that's entomology, it's very close, but epidemiology is really, this is a simple definition of epidemiology. So epidemiology is the study of patterns of health and illness at the population level. So we're not looking at individuals, and we're not doing clinical research or working with individuals in schools. We're really looking at groups of people and trying to understand and identify factors that influence disease or positive health in groups of people. So it's very much a public health discipline, and we're taking the results of our epidemiologic studies to inform the prevention strategies, public health prevention strategies. So again, strategies at the group level. But ultimately, the whole point of this is to lead to optimal treatment approaches at the individual level, okay? So we're starting at this group level, and ultimately the findings that we discover will be implemented, we hope, in the clinic when we're working individually with people one-on-one. So it's really pretty simple. There are 3 basic steps for being an epidemiologist. The first step is to define the subject that we're studying. What is it? What is autism? In this case, I'm going to be talking about. And then we describe what we know about the thing that we're studying. So it's all answering all of these questions. How many people are affected? Who is affected? Where are they? Where does it occur? And when does it occur? What are the trends over time? What's happening today, and how does that look different or similarly to what happened 10 years ago? And what do we think will happen in the next 10 to 20 years? So that's step 2. And after we've done that describing, then the third step is we analyze. So we say, why does this happen? And what are the factors that are causing this outcome? And how can we identify particular factors, risk factors and protective factors that we can relate to this outcome that we're studying that we then can intervene on? And that's the whole point, is we want to identify eventually modifiable risk factors so that we can get in there and intervene and change the ultimate outcome. Okay, so the first step, define. What is autism? Well, what I've listed here is the DSM-5 criteria for autism. The DSM-5 is the diagnostic manual that charts out the definition of many different mental health disorders. And this definition is the latest definition that was published in 2013. And it basically says this, that autism, or autism spectrum disorders, which is what we're calling autism now, consists of persistent deficits in social communication and social interaction across multiple contexts, so that you might observe some difficulties in social communication at home, but you also want to observe it at school and maybe in the workplace, so in many different contexts. There's also a pattern of restricted or repetitive behavior, very narrow interests, and deep interests or activities. The symptoms must be present in the early developmental period, and that the symptoms cause clinically significant impairment in social, occupational, or other important areas of functioning. And finally, that these manifestations are not better explained by intellectual disability or intellectual developmental disorder or delay. So that's the current definition of autism. And as I said, now the terminology is autism spectrum disorders. So all of the previous autistic disorder, Asperger's syndrome, pervasive developmental disorder, not otherwise specified, those were the 3 distinct categories in the last diagnostic manual. Those have all been lumped together under this umbrella of autism spectrum disorders. Now, as I think most of us know, autism has many different manifestations. And what I've shown here is this lovely drawing that my autistic nephew drew for me when he was about 22. He's currently 33. And what I love about this drawing is that it describes, well, these lovely playful animals. We can tell that these are all animals. They're all distinctly different colors, different shapes, different sizes. They're doing different things. Some are on top, some are on the bottom. So they're all really uniquely manifesting here on this drawing. But they all hang together as animals. I think everybody can recognize that. And this is sort of a very a good description for me of what now the field is really calling the autisms. We know it's not one, it's a big spectrum of disorders, many different ability levels in this spectrum. And so it's a complicated spectrum with many, many manifestations. And the other thing that I really love about this drawing is that it illustrates some of the core features of autism in that Eitan, my nephew, has been drawing these animals ever since he was a very little boy. He still draws these animals in different configurations, and he makes lovely artwork that I think is available on cards that you can get online. He set out, I asked him, Eitan, could you draw me a picture? I want to use it in my work and make a poster. We were doing a study of pregnant women and following their babies, and I wanted to have a poster that we could give to the babies when they turned 1 as sort of a thank you for participating in the study. And so he drew this wonderful picture, but he was about halfway through, and we were on a family vacation, and we were ready to go to the beach, and I said, you know, come on, Eitan, it's time to go. And he became very agitated and very upset. And he told me in his limited English that he was drawing 71 animals, and he was only at about, I don't know, number 22 or something, and he really needed to finish before we went to the beach. And so, you know, we allowed him to finish drawing, and that was just a very common episode with him, which was that he had a fixed idea of what he wanted to do. He was drawing 71 animals. He became very agitated when he was interrupted, and it was a good example of sort of this repetitive and fixed and restricted interest that many people with autism have. So this is you know, to sort of keep this in your mind, that when we talk about autism spectrum disorders, we're talking about lots of different things, all that share these core features that I've described. So now on to describing autism. I think, as we all know, the prevalence or the rate of autism, the number of people being diagnosed with autism has increased really dramatically over the past 2-3 decades. And currently, so when it was first described in the first epidemiologic study was in the mid-60s, and the rate then was 4 or 5 individuals per 10,000. And today, the latest estimate from the Centers for Disease Control, there's a surveillance program that's been going on for years and years called ATOM, and their latest estimate is that 1 in 59 children, these are 8-year-olds, carry a diagnosis of autism spectrum disorder. So that's an incredibly dramatic increase over a fairly short period of time. We're still trying to understand what this increase really means. Is it real? Is it artifactual? Is it because the diagnostic criteria has expanded so much over these 20, 30 years? There's better awareness, there's more services, thankfully, some better services in some areas. Or is some of this increase really a true increase in incidence? That is it really many more people today having autism than they did 30 or 40 years ago? And you know, this is probably the most common question I get when people understand what I do as an epidemiologist. They say, well, is this increase real? And what's causing it? And my answer is, you know, we don't really know. There's been a lot of research trying to understand what this is all about, and we don't really know. I think the best estimates are that maybe 50% of this increase can be explained by many of the factors that I've just mentioned. but about half or maybe more of this increase may be a true increase in incidence. And so our job is to try to understand what is driving that. Why do we see more people with autism today than we did many years ago? There's several factors that are fairly consistently found across many different epidemiologic studies that are related to autism. One of the most consistent finding is that there's this very interesting skewed sex ratio where there's about 4 times as many males affected as females affected. And that estimate, depending on the level of functioning of the individual, sometimes that ratio goes up to maybe 5 or 6 or 7 to 1, boys to girls. The more mildly affected or higher functioning individuals, it tends to be more skewed toward males than females, and sometimes the more severely affected, especially those with associated intellectual disability, may have a lower sex ratio, fewer boys to girls, still more boys to girls, but maybe more 2 to 1 or 3 to 1. But that skewed sex ratio is something that's very, very consistent and has been described since the very first time autism was reported, which was in the early 1940s, the first publication. We also know that autism affects people of all races and ethnicities and is seen all around the world. There's more and more studies now looking at autism in different parts of the world, and in places where people have been able to track numbers over time, you also see this dramatic increase. So that's consistent as well. We know that autism affects people of all different socioeconomic statuses, and interestingly, the results are sort of conflicting depending on where you do the study. In the United States, it's been pretty consistent that it's people of higher socioeconomic level that are more likely to have autism in their families than people of lower socioeconomic status or lower income or lower education. But in the Scandinavian countries, sometimes we see the reverse trend, that you see the people of lower socioeconomic status having a higher rate or no difference. So that's a little bit, something food for thought and trying to understand what is this socioeconomic status variable really measuring. And there's also pretty consistent evidence that babies who are born prematurely and twins are much more likely to have autism than babies born at term or born as singletons. So those are some of the general features that are pretty widely accepted and agreed upon in terms of factors that are associated with autism. Okay, so I've gone through steps 1 and steps 2. See, it's fairly easy, right? And now we're going to move on to the last step, analyze, and I'm going to spend most of my time here. So when we talk about doing analytic epidemiology, what we're really talking about is investigating relationships between 2 things, between exposures and between outcomes. And in this case, we're talking about the outcome is autism or autism spectrum disorders, and the exposures can be really anything, any factor, any socio-demographic factor, what we typically think of as environmental factors. An environment, using the broad sense of environment, could be nutritional factors, could be medications, could be health factors, could be toxic exposures, could be lifestyle. So anything that falls really outside of the realm of genetics, and that's another type of exposure. We don't usually think of genetics as an exposure, but it's a factor. We're trying to relate, how does this factor relate to that outcome? And in the history of autism risk factor and analytic epidemiology for autism, there's been a long history that has really been a transition from the beginning, when autism was thought to be a result of lack of parental love, or the so-called refrigerator mother. This was just this horrible concept that was promulgated in the 1950s and the early part of the 1960s, that the explanation for autism was given that the parents, and especially mothers, lacked affection and warmth toward their children, and that was the cause of the isolation and the autistic behavior. Well, fortunately, in the early to mid 1960s, that notion was thrown out the window. There was an understanding that autism was really a biological disorder, and there were biological underpinnings of the behavior that we call autism. And that really started many decades of research into biologic factors. For the first several, well, probably 2 or 3 decades, the research really focused on genetics. Everybody was looking for genetics and doing all sorts of studies. And actually, autism has been found to have a very, very strong genetic underpinning. There have been all different kinds of studies. The interesting ones, I think, that really highlight how we know autism has a strong genetic underpinning is that people started looking at twins, and they looked at identical twins and fraternal twins. And the identical twins, there was a much higher chance of both twins in an identical twin pair having autism than twins in a fraternal twin pair. And what we know about twinning, identical twins really share their genetics, share their DNA, and fraternal twins share half of their genetics. And so when you have a strong concordance, we call it, but when both twins had autism and they share their DNA, then that tells you that, okay, there's got to be something that they probably share genetically. So, and there are many other kinds of genetic studies that have illustrated that there's a strong genetic underpinning of autism. But genetics doesn't explain everything, and actually far from it. And so then non-genetic studies started to be done, and people looking for factors that were outside of the genetics, and now there's a very strong recognition that autism is really a combination of both genetic and non-genetic factors. And it's probably a combination, many different combinations of genetic factors and non-genetic factors that are occurring at the right or the wrong time during development that lead to this manifestation of autism, or the manifestations, because autism, as I hope made clear, is not just one condition. So in terms of the prenatal origins of autism, there's a lot of evidence that now leads us to know that autism really begins very, very early on in the developmental period. There's a whole host of studies looking at the brain that have found different brain anomalies in different parts of the brain that if you know anything about the development of the brain, those certain anomalies in the brain indicate the timing when that insult occurred, timing during gestation when that insult occurred, and so that we know, for example, Purkinje cell loss, that was one of the very first neuroanatomical findings. Purkinje cells are a certain type of cell in the cerebellum, and that loss had to occur, that insult had to occur prior to 30 weeks gestation, because that's when that develops. There's all different kinds of brain anomalies that have been found, and it's indicated to us that these brain structures, because of when we know when they develop, that when they're developing in an unusual way, had to occur during the gestational period. We also know that there have been many studies looking at infection during pregnancy and women who have had infections. One of the classic examples was of a rubella outbreak, and women who were exposed to rubella during pregnancy, there was a much higher rate of autism in the children born to those pregnancies than would be expected. And there have been many, many studies. Actually, some of the very earliest epidemiologic studies have looked at infection in mom. around the time of pregnancy and found increased rates of infection among the women who had autistic children compared to women who did not have autistic children. And I'm going to say a lot more about infection in a little bit. We also know that there have been many studies looking at obstetric complications, so complications during pregnancy and during delivery, and either as a constellation of issues or one by one, for example, cesarean section or preeclampsia or many different kinds of complications that can happen during pregnancy. And many of those factors have been associated with an increased risk for autism as well. And finally, there have been many studies looking at medication use of moms during pregnancy and have associated those medications with increased risks of autism. Now, they're not dozens and dozens of studies looking at these factors, and the evidence isn't clear enough to say, okay, we know this causes autism, but these are associations. And this is another thing that I want to make clear, that in epidemiology, we're looking at how factors associate with outcomes. We really never get evidence that says this factor causes that disease or this outcome. But it's study after study, using different study designs, looking at different study populations, and asking similar questions. When you get similar answers, then that leads us to be able to conclude that, yes, this factor is strongly associated. And then we have to look underneath all of that and look at the biology and see what's actually going on in the body and at the sort of molecular level or at a smaller level to understand how this factor could be impacting and creating this outcome. So there's a lot of evidence that, as I said, that tells us that the etiology of autism or the causes of autism are really twofold, both genetics, genes, and non-genetic or environmental factors. And it's really the combination of these factors that are occurring at critical exposure windows and in very early development, sometime in the very, very beginning of gestation and all the way through, that are the important factors. And it's likely that different clinical subgroups or the different kinds of manifestations we see across the spectrum, they likely have different combinations of these genetic and environmental factors. So we're talking about a multifaceted, very complex set of conditions, both the risk factors we're looking at across the spectrum, and the outcome is this big spectrum, and so it can get very complicated very quickly, and very challenging. So here's just a sort of a conceptual model of autism etiology that hopefully summarizes what I've just been talking about. We've got environmental factors that could act either directly or indirectly and on the outcome of autism. And first of all, autism, represented here by ASD, is really complex in and of itself. It's behavioral, developmental, and medical issues. And what I didn't mention already, but I will now, is that in addition to the core behaviors and conditions and impairments that define autism, there are many different co-occurring medical and psychiatric conditions that can take place as well. Not in everybody, but there's a whole range of other mental health conditions, anxiety, depression, and many others that many individuals with autism experience, especially as they get older into teenagehood and adulthood. And there's also a lot of different medical complications that may co-occur with the autism itself. So we have environmental factors, we've got genetic factors, and then we've got a whole host of factors in the middle there that are maybe influenced by the genetics or the environmental factors. So immune function and hormonal function, metabolic factors, and this whole field called epigenetics, which is really fascinating. And all of those, either in combination or individually, can impact the outcome and result in what we call ASD. Okay, so now I'm going to turn to illustrating some of this and how we go about studying some of these factors. And I'm going to concentrate on the immune system. So the immune system is our body's natural defense mechanism. It detects a wide variety of foreign agents, and the immune system is responsible for keeping us healthy when we encounter infections or other kinds of exposures of foreign agents, so to speak. And the nervous system is this very complicated system, includes the brain and the peripheral nervous system that transmits signals between different regions of the body to make our bodies function. And it's a very sophisticated set of interactions between these complex neural pathways. And so then, you know, for a long time, the immune system and the nervous system were thought to be completely independent, but we've learned that they're very, very connected. And in this whole field of neuroimmunology, which really talks about the complex interactions between the two systems. And these systems are interacting both during regular resting and normal conditions, and they're also interacting in response to injury and also very strongly during the developmental period. And some of the factors that are involved in this interaction are these molecules called cytokines and chemokines, which are basically molecular messengers. They're messengers reacting to one set of conditions and communicating to the body to do another set of functions to keep us alive and healthy. So the cytokines and chemokines are signaling chemicals, and there's also all of these pathways, immune signaling pathways, and antibodies also as well, which are our defenses against foreign invasion, among other things. So why look at immune function in autism? Well, there have been decades of research that have looked at various aspects of immune function from different angles, and have really, when you look at the body of evidence combined, point to a very strong connection between immune system function and immune system dysregulation and autism. From the genetic studies, there were many genes that were found that to be more present in individuals with autism, and these genes regulate immune response. So I'm talking about now associations in people with autism themselves. People had looked at different markers, molecular markers in blood that was taken from people who already had a diagnosis of autism, and found abnormal levels of different immune molecules, different antibody levels and different cytokine and chemokine levels in children with autism versus children without autism. There have been different kinds of activities in brain and cerebral spinal fluid. These are different, not blood, but in different organs in the body that also looked at immune function and found differences between individuals with autism and those without. And also reports and studies that have shown that children and individuals with autism have increased rates sometimes of infection or different immune conditions like asthma and allergy. Now, I want to point out that the studies are not all consistent in the specific molecules that have been found to be elevated or lowered, or the specific clinical conditions that are found to be more associated with autism than not. But when you look at the body of evidence, there's always there's a very consistent pattern that immune system function is related in individuals with autism. But the question is, are these immune changes a consequence of the autism and the autism behavior, or are they preceding the autism? Are they somehow related to causing the autism? So timing is really everything. And so we decided to try to tackle this issue by looking at mothers. and looking at the timing of these insults. When we, do we find changes in the mothers of children with autism in terms of different autoimmune or immune conditions, or even in the immune, when you measure blood or measure the blood taken from women who have had autistic children, do we see any differences in the blood? And so when we concentrate first on the postnatal period, we have looked at immune markers, or I say we, but the field has looked at immune markers in blood that was collected after a child was diagnosed. And one particular kind of study has looked at autoantibodies, and these are antibodies that are generated to. An autoantibody is something that's generated that sort of attacks self, so that's autoantibody, and it's an antibody that's excuse me, targeted to something that's going on in the person's body. And in this case, the antibodies were to fetal brain proteins. So these are proteins that are in the brains of fetuses that are developing in utero. And what was found in this study, and this study was done by my colleague Judy Vanderwater and colleagues at UC Davis, what was found is that the blood from moms who had had an autistic child looked a little bit different in some of these, not all mothers, but in about 20%, looked a little bit different than the blood, what was found in the blood of moms who had typical children. And okay, so the TD stands for typical development. This is, AU stands for autism, and DD is developmental delay. So these are kids without autism, but some other kind of developmental disorder. And what you can see, first of all, this is a Western blot, and everybody has these dark bands here and here. So those are just sort of put on there as standards. You're going to measure that in everybody's blood. But what was found to be different here is that the children with autism had some extra dark bands here and here that we didn't see at all in any of the kids with typical development. And this band pattern was very specific to autism. As I said, about 20, in this study, I think it was about 11% of the kids, of the moms who had autistic kids, had this band pattern, either one or the other, or a combination of the 37 and the 73, and none of the moms of the typically developing kids had these bands. So this was, again, taken from blood after a child was diagnosed. Now, what we really would like to know is what's happening during the critical period of brain development. So this would be before the child is born, and while the child is in utero and developing, and when the brain is developing, what's happening in that environment. And so we've done many different studies looking at this prenatal period, looking at maternal conditions that have to do with immune function, so looking at maternal infection, asthma, allergy, autoimmune diseases during the time of pregnancy, and also looking at patterns of these inflammatory markers, or these cytokines and chemokines, in prenatal serum. So this is blood taken during pregnancy to see if we can find any differences in those samples in women who eventually go on to have a child diagnosed with autism compared to women who have children who are typically developing. And also we've looked at autoantibodies, and we and many other people have looked at this as well. There are, I'm going to flash here, many studies on looking at infection around the maternal infection around the time of pregnancy. There have been many different studies done with many different study designs. One thing that every different study design has advantages and disadvantages. Some of these studies were based on surveys of populations of people coming into a particular clinic. And so they relied on women to report and think back on, and sometimes this is many, many years in the past, what their experiences was during pregnancy, and did they have an infection, and what kind of infection, and when was it? Was it the first trimester or the second trimester? And as you can imagine, there are many problems with recalling events like that happened sometimes so many years ago. So the accuracy of those kinds of studies is sometimes problematic. There have been many studies that are based on registries. For example, registries either from a population of an insured population, such as the one I work in Kaiser Permanente, where it's not so much a registry, but a whole population who's getting health care in a certain system, and their interactions with the healthcare system are documented in a medical record, and you can go back and look at those medical records and sort of count up who has an infection, who doesn't have an infection, and link that to the outcome of the children. There have been many studies from the Scandinavian countries, which are whole population of the whole population living in that country or in a specific region of a country, that in the Scandinavian countries, the people are linked, have an individual unique identification number that is recorded in many, many different databases, and health databases, and in school databases, and in insurance databases, and employment. And so you can link all of these different, very rich databases together to make connections between people's health status in a certain period of their time and the health status of their child. And many of the studies looking at infection have been done based in those Scandinavian studies. And so there are pros and cons of all these different studies, but what's missing often is direct measurement of what's happening during that critical period of brain development when the baby is in utero, and what's actually happening on a molecular level. So with maternal infection and autism, while there's a lot of evidence that points to a connection, there's still many unanswered questions. There's disagreement between the studies about which type of infectious agent it is. Is it a viral infection, a bacterial infection, a parasitic infection? What's the timing that's most critical? Is it early in pregnancy in that first trimester, or very late in pregnancy around the time of delivery? And is it the actual agent, the infection itself, the virus, let's say, that's the actor that's somehow dysregulating the brain development of the baby? Or is it the immune response to that infection, the inflammation associated with that infection that may be measured by fever, for example? And there's some very exciting research that's coming out now that's really pointing to fever and the inflammatory response that may be the critical agent here that's acting, not so much the infectious agent itself, but maybe the response to it. But these are still questions that are unanswered completely, and which studies, more studies are needed to look at. And so, likewise, in terms of maternal autoimmune diseases, there have been many studies done looking at a history of autoimmune disease, like rheumatoid arthritis or MS, many different thyroid autoimmune disease that have been found to be associated with an outcome of autism in the children, and maternal history or a family history. But again, there's a lot of disagreement about which particular autoimmune disease it may be, and if it's specific to autism or more specific to, or more general to developmental disorders in children. And again, the timing of these diseases and when, is it the immune response again or the disease itself? That's the actor that we should be most interested in. These are still unanswered questions. And also there are questions about, well, maybe there's some similarity or some common underlying susceptibility, that there may be a genetic association with some of these autoimmune diseases that may also be the same genetic association with autism. So one particular cause, if you will, ending up with two different outcomes, and that that may be why we see an association between autoimmune diseases, and autism. So still many, many unanswered questions. So we decided to mount this study that I call the Early Markers for Autism Study, or EMA. This has been a study that's been going on for many, many years. And it really was designed to overcome some of the limitations, and I say some of because it couldn't overcome many of them, but overcome some of the limitations of previous studies. And the really interesting thing about this study, and I think we were in a unique position to do this study because California has many resources that are already there, present, that we were able to get permission to use and to link different databases and different specimen registries together to pull together information on a group of mother-baby pairs to look at many of these questions and really interrogate what was going on during pregnancy of these women who ended up having children diagnosed with autism spectrum disorder or diagnosed with another developmental disorder, or had children who were not diagnosed with any developmental condition, but they were, as far as we could tell, typically developing kids. So this study, the EMA study, is investigating early biologic markers of susceptibility and exposure from critical periods of fetal brain development. So that's during in this case, we had biospecimens from the second trimester. And we were able to look at both genetic factors and these immune factors and many other environmental exposures that we could measure in these blood samples that we had. And then bring all those different factors together. So the way we did this is called a population-based case control study of mother-baby pairs. And what we did is in California, there's both prenatal screening of moms during pregnancy for a variety of different conditions and newborn screening, which in every state there's newborn screening. But in California, these newborn screening blood spots, which I'm showing here on the bottom right, are saved. After the newborn screening testing is done, the remaining blood spots are saved in a huge freezer, which sort of looks like Costco. You know, it's rows and rows and rows of boxes. And we have about 500,000 babies born a year in California, and this repository has been present since 1980. So you can imagine, I can't even, it's a lot of blood spots. So what we did is we said, okay, we want to identify children who have autism and identify children who don't, and we want to identify these children for whom we can get both a newborn blood spot, so we can measure what was going on right at birth. This is years prior to a diagnosis. And also we wanted to identify these children whose mothers had had prenatal testing and whose prenatal blood after the testing was saved in an archive. And in California, for some years of time, in a certain region, the leftover blood from prenatal testing was also saved in another freezer, not quite as big as the Costco freezer, but another freezer. And so we were able to identify these children first and then go back and link them to, almost everybody has a newborn blood spot, but not everybody had a maternal sample during pregnancy. But we identified those children for whom both samples were available. And so it's a retrospective study, but we're using prospectively collected specimens. And we didn't have to do any new data collection for this study, because all of the data that had to do with the diagnosis and all of the samples were already present, and some of the information that was collected when the samples were collected. What we had to do was then measure what we wanted to measure in those samples. So it was a really pretty interesting and very efficient way of doing a lot of science without having to go and recruit lots of women or collect blood during pregnancy and then wait many years to see who ends up with autism and who doesn't, and that's a very expensive proposition. But to do this sort of retrospective cohort study, or is it actually a case control study, because we're comparing cases of autism to controls, it's a very efficient way to do it. So this study was done in 2 phases. We had 2 separate pots of money. This was funded by the NIH. We've looked at many different things in these blood specimens from the babies and from the mothers. And just to use this diagram again, we've looked at many different environmental exposures, organochlorine pesticides and hazardous air pollutants by linking to air pollution databases. We've measured different flame retardants and perfluoroalkyl compounds in the blood. These are all different kinds of chemicals that there was some good science suggesting that they could impact, early exposure could impact the development of the brain. We've looked at many different kinds of genetic markers, and then we've also measured many different kinds of immune molecules in both the mom's blood during pregnancy and the baby's blood at birth. So I'm going to show you some of the results just as sort of at a high level. In the first phase, we looked at the maternal antibodies. Now, if you remember what I showed you before, I showed you a slide that looked like this, but what I was talking about then was blood that was taken from moms after her child had a diagnosis, and sometimes many years after. And the question then was, well, okay, that, you know, there was a timing issue. Well, that's what's present in her blood now, but if the child's already diagnosed, we don't know if what we're measuring now after, you know, when the child's, let's say, 5 years old, looked anything like what was happening in her while she was pregnant with this individual, with this child. And so this study and this slide is looking at prenatal blood. This is the blood taken from mom when she was pregnant with this child who was later diagnosed with autism. And what you can see basically, it's the exact same picture that we saw before. So in the prenatal blood, we saw that there were these bands, protein bands at 73 kilodalton and 37 or 39 kilodalton in the mom's blood for the autistic children, but not in the control moms. So it's a very, very similar pattern. So that was very interesting because we said, okay, we found that this is something that was present during pregnancy, and it's probably and we don't know exactly when it arises. It could happen way before the pregnancy, or it may arise during the pregnancy. We don't know that answer yet, but we could time it. The timing was clarified a little bit. The other thing we measured is cytokine profiles, and that these cytokines are these molecular markers and messengers, and we found that there were some elevated levels of some of these cytokines in the case mothers compared to the control mothers. So there were some significant differences there. We also found that another cytokine called IL-6 was elevated in the mothers of the kids with developmental delay, but not in the mothers of the general population or the typical controls or the autistic children. So we're finding some differences. That's the point of showing you these slides. And the differences that we found in the autism moms was consistent with this allergy asthma phenotype. So that went along with our clinical data showing that asthma and allergy during pregnancy or around the time of pregnancy was more common in the moms who had an autistic child. Okay, in the interest of time, I'm going to kind of go through this. This was the second phase of EMA, and it's just to show this is looking at these same cytokines and chemokines in a whole separate group of mothers, also showing differences. In this case, we didn't find overall differences between the autism sample and the general population sample, but we did see very different, striking differences, which you can see in red in that last column between moms who had kids with autism with an intellectual disability and moms who had a kid with autism without an intellectual disability. So it's suggesting that there's maybe some molecular or differences between those two phenotypes of autism that we have to look into more. So we're doing many more analyses of these data. We can look at just about every combination. These are some of the data that I've already showed you, looking at the immune samples, the immune molecules, and the autism. but we've published, and Kristen Lyle has published 3 papers looking at these environmental exposures, and we found some interesting results there, showing associations with some exposures to these chemicals. We've looked at genetics and autism, and then we can look at combinations of the environmental factors, the immune factors, or the genetic factors and the immune factors, or all 3 factors. So there's many ongoing papers that we're writing right now and analyses we're doing, and this is this is showing some results from looking at the genetics, and with my colleagues at UCSF, Dr. Lori Weiss and colleagues there. And what we found here are some very interesting things which basically show that the cytokine level in the mom during pregnancy, yes, it's controlled by maternal genetics, but it also looks like it's controlled by by fetal genetics, which is a little bit hard to understand, but it's very interesting. And at the bottom, what you can see is that there's certain level of one of the cytokines was found to be controlled by a maternal gene, but also by a baby gene, which was a different gene. So there's interaction between the fetus and the mom, and it's going in both directions. A lot more work to be done there. So, where do we go from here? Well, there's clearly many, many more questions to be answered, and I'll just go quickly. We are currently looking at data in our Kaiser population, and the Kaiser Permanente population in Northern California is a very wonderful place to do autism research, because we have this research unit embedded in this healthcare delivery system that has a huge membership. It's over 4 million people, with a very diverse membership, race ethnically and socioeconomically. We have both government insured and privately insured members. We have this fully electronic health record, so all of the interactions of patients with the health system, including telephone calls and office visits and lab results and medications, are recorded in the electronic medical record. And the population is very representative of the greater population that lives in this geographic area. This is the San Francisco Bay Area and the Sacramento area. So it's a really huge, diverse population to study. I've been tracking the autism prevalence in Kaiser for the last 15 years or so, and the most recent data that were from last June show the numbers and the rates of autism, and they pretty much track with the national trends. In fact, our numbers are a little bit higher. The 1 in 59, 1 in 57 figure, or 1 in 59 figure is about a 1.7 or 1.8%, I think. And in 8-year-olds, and in our 5 to 9-year-olds, we have about a 2.6% rate. So we have 3 diagnostic clinics that are specifically autism diagnostic clinics in the region. And kids are given a very complete workup and with some standardized instruments. And so we're catching all of our kids very, very early, actually. So so we have a very large autism population within our membership. We also have this pregnancy cohort, which is a collection of, we invited women who are coming in for prenatal care to join this project, and basically we asked them to donate some blood from when they went to the clinic to get their prenatal testing. We asked them to donate an extra vial or 2 of blood, and also to complete a short survey online, and to let us have access to their medical record information. And we enrolled about 25,000 pregnancies, so it's very large, and about 20% completed the survey. And almost 80% of these participants gave a sample of blood both in their first trimester and their second trimester. And the amazing thing about this collection is that not only is it large and diverse, but it's very representative of the general population of women coming in for prenatal care in our system. So it's very generalizable. And so I wrote a grant and got funded to do this impact study, which is following up on all of the findings that we saw in EMA and other people have found, looking at immune function during pregnancy and trying to understand, looking again at what's measured in the blood of moms, but not only in the second trimester, but first and second trimester, so we can look at trajectories during pregnancy. And we can also link those data to clinical manifestations of what the mother is experiencing and what she's doing during pregnancy, data which we did not have in the EMA study. And so this is a true longitudinal prospective study looking at this gestational inflammatory conditions and genetics and how that may impact on child development. And so this is just sort of a picture of what we're doing. We're overcoming many of the limitations we had in the EMA study by having both samples during pregnancy and a couple times during pregnancy, linking that to this very deep, rich information about maternal clinical conditions during pregnancy and before. And then all of these babies were born in Kaiser, and we've been able to follow them through their medical records from birth to when they get diagnosed, and look at any kind of outcome that these children have. So not just autism or a set of developmental disorders, but the whole range of neurodevelopmental conditions. So we can try to look at the specificity of these different different molecules during pregnancy and different conditions during pregnancy and what the outcomes of the children are. And also look at maternal genetics too. So it's a very exciting study. We're just in our first year. It's a 5-year study. This stuff takes a lot of time, unfortunately. But we're hoping that it'll really shed new insights into this whole area of maternal inflammation. And, why is this important? Well, again, we're starting at the population level, looking at health conditions and patterns in mothers and children so that we can understand associations and eventually get to the individual level and use all of this information gained to tailor interventions and prevention strategies at the individual level. So that's an application of our public health strategy. And with that, I'll end, and I just want to acknowledge my wonderful team. We all love to eat and cook, and so here we are. And my close collaborator, Dr. Judy Vanderwater at UC Davis, who I've done much of this work in collaboration with. And also, finally, many of my funders. So thank you for your attention, and I look forward to fielding your questions. Thank you. Yes.
I'm not sure if you stated this before, but I was wondering if for positively diagnosed autistic children, if there was a difference in prenatal cytokine levels and post-born cytokine levels.
Let me see if I understand your question. So if there's a difference in what we measure prenatally to what we measure postnatally, is that what you're asking? Yeah, well, the prenatal cytokines we're measuring in the mother, and postnately we're measuring in the child, and they're different immune systems, and they're not correlated. In fact, we've looked at in a small sample those cytokines we measured in both the mom and then the baby at birth. So this is before the child was diagnosed, and there's not a whole lot of correlation. They're different.
Hi, thank you very much. I have a question. I know that your study focuses on maternal infant work, but I'm wondering if the markers don't indicate the mother, do you take a look at the father if he's present?
Yeah, there are many, that's a good question. I'm glad you brought up the fathers. Fathers are a very important part of this equation. And there are many studies that we're doing other studies looking at fathers and fathers' genetics and fathers' exposures and so on. In terms of, and for example, the autoimmune diseases, many of the studies have looked at, including one that I've studied, looked at maternal autoimmune diseases and paternal autoimmune diseases and just sort of general family history. In our study, we did not find an association with a history of autoimmune disease in the dad, but we did find something in the mom. And, you know, during child development, the environment that the child is in is the maternal environment, not the paternal environment. But, you know, there are many reasons to look at paternal factors and association with what the child's outcome is. So thank you for bringing that up.
Hi, you mentioned the autoimmune as well as an infection during pregnancy. I'm wondering if you've looked at possibly, because myself having a child with autism and being immersed in maternal child health at the time, you know, there was a overwhelming belief that things like Tylenol was safe. And you lost, you know, with this autoimmune allergy versus infection, you know, when you combine the two, the only thing they would tell you that was safe during that time was Tylenol. And years ago it was prescription only, and then it became over the counter. Just wondering if you have looked at another variable, I hate to say it, how people actually treated the symptoms of the allergy or the infection.
Yeah, very good question. So treatment, yes, it's very important to look at both the infection or the allergy and medications that people are taking for it. And we've tried to do that in our analysis, so we have information on both the you know, let's say the autoimmune disease and some medication that someone or let's say asthma and something that a woman has taken for the asthma. And when we've done that, we haven't seen, our data have shown that it's really the underlying condition and not the treatment that is related to the increased risk. Now, having said that, you know, that's not there's a lot more research that needs to be done, and this is an area that's extremely difficult to tease apart those two because they happen together, and it's also sometimes from medical records difficult to tell what particular medication was given for what particular condition of the mom. So we're doing a little bit of guesswork, and again, this is an area that needs a lot more work to try to tease apart those two factors.
Just a quick follow-up, but the anti-inflammatory effect is not present in Tylenol. There seems to be a connection in some way with that, you know, with many of the variables that you've been talking about. Just curious.
Yeah, so there are a few studies that have looked at with fever, looking at how the fever or the infection was treated with an anti-inflammatory or with a non-anti-inflammatory. So, for example, with Tylenol or with Advil, and trying to tease apart, you know, try to isolate, is it the inflammation or is it maybe the condition? And the data are pretty sparse in this area. I know of a couple studies that have looked at that. We're going to try to look at that a little bit more. It's hard to, you know, to have all of the data with a large enough sample size, a large enough population to study to really be able to do justice to that topic. But I know that there are ongoing investigations in that area, and it's a very important question.
Dr. Krone, thank you for your really beautiful talk. My question relates to when you're looking at risk factors such as inflammation or autoimmune or infection or possibly toxic factors that a child has been exposed to or maybe prenatally was exposed to. Have you had any association between the rise in these factors and the rise in autism?
Mm-hmm. Right, so that's, you know, one of the reasons why people started focusing on environmental factors is because we knew that there was a strong genetic component, but people say genetics don't change that quickly. And so maybe there's something in the environment that's changing at the same rate or in the same trajectory as the increasing numbers, the rise in autism. And so, their attention was turned to many factors that may be newer in our environment now that maybe weren't present 40 years ago, that also have a strong biologic, there's a strong possibility that these particular exposures there's plausibility that they could impact on brain development. And so many of the chemical exposures that people are looking at have both, they're more commonly used now than before, but they also have, and I want to make a point here, that they also have some biologic plausibility. There are many factors that have increased over time, like people driving, electric cars, for example, that follow the same trajectory in increasing rates as, let's say, autism. But there's no biologic plausibility that those two factors, for example, could be connected. So we have to be very careful when we're looking at these sort of trends, to look at also time trends, but also what is the scientific evidence that this particular factor could impact this particular outcome that we're interested in. Mm-hmm.
Thank you so much for the work that you're doing. I'm the mother of a 20-year-old son with autism, and I'm just very excited by the leaps and bounds that have happened really in the last 20 years, so thank you. I have a couple of questions, but one is related to looking at environmental issues, again, kind of piggybacking off the last question, but I wondered, have we looked at issues surrounding cell towers and some of the more magnetic wavelengths that are happening in our environment? Is that something that we're looking at now?
That's something that I am not looking at, and I'm not really aware of. Maybe some of the other epidemiologists know. They're shaking their heads. So I don't think there's any work that's going on in that area. I know that's a huge change in our environment over the last 10 years even. And I think there's work, people have looked at other kinds of outcomes, I think some cancer outcomes, but I'm not aware of any association, any studies looking at that and neurodevelopmental outcomes.
Thank you so much.
I think there's... I think there is one more question.
This is a bit of a hypothetical. Could you just riff on the fact that if you found out that some of these things that you're looking at are for certain in terms of a risk factor, what would that look like in terms of public information and helping folks, with pregnancies and that? Could you take one of those factors and say, if this turns out to be the pearl of greatest price, this is what we would be telling folks?
Yeah, well, that's a very difficult question. That's a very important question. So I'm not going to sort of off the cuff give you the public service announcement, because that has to be thought through very, very carefully. And I think communicating these kinds of results to the public, even me standing up here and talking about these factors, knowing there are many parents in the audience who have autistic children, and what you might be going through your minds when I'm saying, oh, maybe it's maternal infection, and maybe it's this, and maybe it's that. It's a difficult thing for me to do, and it's a difficult thing for people to receive, because what's the actionable item, or what can I do now? Oh, great, now you tell me that. What can I do with that information? So it's very important to think about how we communicate these results and what we do with the data. And I think the important thing is to understand that it can be very difficult to hear, and to give people something that they can actually do, or maybe not do for this particular child, but do for future pregnancies if people are contemplating having additional children, and what we can do in terms of prevention. And for example, maybe one day these results will be solid enough to then lead to some kind of prenatal screening, if people choose to have prenatal screening, that could at least identify maybe pregnancies that are at risk, so that interventions could be started very early on in the child's life to prevent some of the difficulties that people might have later on. So to make lives better, for example, or if people choose to take some actions to prevent an outcome, that would be their choice. So that's the kind of thing I'm hoping that can be done with these results, but the communication around it is extremely difficult, and we need to engage, our communication experts along with our clinical experts and our research partners with parents to figure out the best way to communicate and to provide information that's going to be positive and, helpful rather than a real bummer and a downer. So that's the best answer I can give you right now. Thank you. Yes.
I found it interesting when you mentioned that in the United States, autism diagnoses are associated with a higher socioeconomic status, whereas in Scandinavian countries, it tends to be lower, knowing that Scandinavian countries tend to have better access to health care, and that in the United States, certain minorities and people of lower social classes have more trouble accessing health care due to time and money constraints. And I'm wondering if you could say something about whether certain groups, certain socioeconomic groups in the United States tend to be underdiagnosed with autism, and what's being done to address inequities in health research. I know that when you talked about your Kaiser Permanente study population, it seems like that's something that you thought about, and then you have a pretty diverse population. But I was just wondering if you could say a little bit more about that.
Yeah, thank you for that question. That's a big issue. So I think in this country, the trends we see are probably partly or maybe mostly driven by access to health services. That's, we don't know completely, but and access, we know that that minority children and people of color and underserved populations, underrepresented populations, people with lesser means are diagnosed at an older age. The children are, compared to more socioeconomically advantaged families. And so with that delay in diagnosis, or the later age at diagnosis, means later access to services and interventions that might be helpful for some of these children. So that's a real problem, and how do we address that? I know that in our system, in Kaiser Permanente, where we have both government-insured and privately insured patients, there's no difference in age at initial diagnosis, which is under 3 years of age now across the board for all different race ethnic groups. So I think that is, one indication of success, that at least when you have access and you have insured, however you get that insurance, if you have access to services, that everybody could be diagnosed earlier. And hopefully we can spread that across the country. It's very difficult with our current healthcare system being, little bits of here and there, and not everybody having access. But yeah, it's problematic, because everybody, no matter who you are, deserves the best care and the earliest identification and the earliest intervention, and that's just not happening right now in many communities.
Thank you so much for your talk. It was, I think, a great blend of big picture and then some of the detailed findings from your work and the work of your colleagues. I want to thank the audience for coming and let folks know that we do put on a few events a year, and if you're not on our mailing list and you didn't sign in when you came in, please do let us know how to reach you so we can tell you about things like our concert in the fall and next year's annual public health lecture. And anybody who's interested in learning more about the Eagles Autism Challenge, we have a table in the back, and feel free to make your way over and even sign up to join a team. So thank you so much, Dr. Krone. We appreciate you.
Thank you very much. Thank you.